Welcome to today's Explainer. We're jumping right into the ultimate biological blueprint. Just think for a second about the incredible microscopic architecture inside you right now. An architecture that dictates human health, biological variation, and really, our very survival as a species. Today, we're exploring the chromosome, the microscopic powerhouse that makes you exactly who you are.
Okay, let's dive into this. Here's our roadmap for today. We'll start with the blueprint of life, take a look at the DNA folding journey, and break down the anatomy of a chromosome. Then, we'll hit dividing and multiplying, look at when things go wrong, and wrap up with why chromosome research matters.
All right, part one, the blueprint of life.
You know, to really get what chromosomes are, it kind of helps to look at the word itself. It's a mashup of two Greek words, chroma, meaning color, and soma, meaning body. So, colored body. Why? Well, back in the day when early scientists were peering through their microscopes, they noticed these thread-like structures in the nucleus just soaked up artificial stains during cell division. They literally lit up with color, and that vibrant pop is exactly how researchers first spotted and started studying them.
Moving on to part two, the DNA folding journey. I absolutely love this part. I mean, getting all your DNA into one microscopic nucleus, it's like biological origami on extreme mode. Let's walk through this folding journey. First up, we've got the raw genetic material, that classic DNA double helix. But it can't just float around loose, right? So it quickly wraps super tight around these specialized histone proteins to form what we call nucleosomes. Next, those nucleosomes coil and fold up onto themselves, condensing into thicker structures known as chromatin fibers. And finally, when the cell gets ready to divide, that chromatin supercoils even more, packing down super tight to form those classic, distinct chromosome shapes. It's fast, it's dynamic, and honestly perfectly orchestrated.
And this brilliantly illustrates that crucial second step of the folding journey. When scientists look at nucleosomes under a really powerful electron microscope, the DNA wrapping around those histone proteins looks exactly like a microscopic necklace. They literally call it the beads on a string formation. It's the foundational level of chromatin organization, keeping our genetic code nice and ordered before it folds up even further.
Next up, part three, anatomy of a chromosome. So, we've seen how they fold. Now let's look at the actual anatomy of a fully assembled chromosome that's holding your DNA right this second. It's got a very specific structure. First is the chromatid. During cell division, a chromosome actually has two of these, sister chromatids, and they are exact, identical copies of each other. Pinning them together is a constricted region called the centromere. It acts kind of like a structural belt and an attachment point for the cell's machinery. Finally, at the very tips, we have telomeres. Think of telomeres like those little plastic caps at the end of your shoelaces, the repetitive sequences of DNA that protect the chromosome ends from fraying or taking damage when the cell replicates.
Now, that centromere we just mentioned, it doesn't usually sit perfectly dead center. Because of that, it splits the chromosome into two distinct sections, or arms. You've got the P arm, which is the short one. P actually comes from the French word petit. And then you have the Q arm, which is the long arm. The relative lengths of these two arms are actually the main way scientists map and classify chromosomes.
Let's see how this builds into our classification system. Depending on exactly where that centromere pinches the structure, we categorize chromosomes into four types. Metacentric means the centromere is dead center, so the arms are equal. Submetacentric means it's a bit off-center. Acrocentric means it's pinched really close to one end, giving it a tiny P arm. And finally, telocentric, where the centromere is at the absolute end. But here's a fun fact. While you'll see telocentric chromosomes in some animals, we humans don't have them at all.
So, what's the magic number for us? It's 46. If you're a human, this number is absolutely crucial. 46 is the precise, specific count of chromosomes found in typical human cells. It is literally the defining number for our species' genetic instruction manual.
Let's break down how those 46 chromosomes actually organize themselves. They don't just float around solo. They pair up into 23 distinct pairs to form what we call a complete karyotype. 22 of those pairs are autosomes, meaning they carry the vast majority of your everyday genetic traits. But it's that final, single pair, the sex chromosomes, that determines biological sex. An XX pairing typically results in a biological female, while an XY pairing results in a biological male.
All right, part four, dividing and multiplying.
We've looked at the static structure, but chromosomes are always dynamic. They go through two totally different types of cell division depending on what your body needs. First, there's mitosis. This is your body's go-to for growth and repair, churning out two completely identical daughter cells. The chromosomes have to replicate flawlessly so every new cell gets a perfect copy. But then we have meiosis. This is strictly for reproduction, creating gametes like sperm and egg cells. The wild thing here is that the cells deliberately end up with only half the normal chromosome number. And even crazier, during meiosis, chromosomes actually cross over and mix up their genetic material just to create massive genetic diversity. So one process demands perfect cloning, while the other is all about beautiful, unique variation.
Which brings us to part 5, when things go wrong.
Now, what's really important to understand here is how these microscopic cellular errors lead directly to recognizable genetic conditions. Because this whole process of folding, packing, and dividing is just so insanely complex, it doesn't always go perfectly. And the human stakes are profound. These abnormalities usually fall into two categories. First, numerical abnormalities, like having an extra chromosome or maybe missing one entirely. This directly causes conditions like Down syndrome, Turner syndrome, or Klinefelter syndrome. Second, there are structural abnormalities. The count might be perfectly fine, but a piece of the chromosome gets deleted, duplicated, inverted, or even translocated, meaning a piece breaks off and attaches to a completely different chromosome. Ultimately, these microscopic changes can result in significant developmental disorders, infertility, or severe genetic diseases.
And finally, part 6, why chromosome research matters.
Let's zoom out from the microscopic nucleus to the big picture and see the real-world impact of all this science. Studying chromosomes isn't just academic, it's the absolute bedrock of modern medicine and biotechnology. Scientists use some incredibly cool, cutting-edge analytical techniques to unlock these genetic secrets. We use karyotyping to visually map out an individual's complete set of chromosomes to spot any of those major numerical errors. Then there's FISH, fluorescence in cytohybridization, which literally lights up specific genes on a chromosome with fluorescent probes to catch structural defects. And of course, advanced DNA sequencing lets us read the precise molecular code right inside the chromosome itself.
These tools are absolute game changers for genetics, molecular biology, and evolutionary biology. But honestly, the most exciting part is looking at what comes next. As our tech to read, analyze, and even edit chromosomes gets better and better, you really have to wonder, what new diagnostic and therapeutic breakthroughs are just waiting to be discovered inside our own DNA? Could we one day correct structural abnormalities before disease even has the chance to manifest? The answers are literally folded up inside you right now, just waiting to be read. Thanks for joining me on this explainer today, and keep exploring.